Hi everyone, welcome to biology practical session. This is experiment 5 and this is one of the question in the exam. So this experiment carries 5 marks, 3 marks will be for slide preparation and 2 marks will be for the result. Now to start with, the name of the experiment is staining of nucleic acid. So here we are going to stain the nucleic acid. Means in turn we are going to stain the nucleus and we are going to see it through the microscope. The aim of the experiment is that we have to make the material, we have to prepare the material in such a way that we are going to observe the nucleic acid in it by staining it. So the main purpose of staining is that we are adding a color to the material so that it becomes visible. Now, before starting the experiment, so it's about the principle of experiment. So we all know that nucleic acids. So nucleic acids, there are two types, DNA and RNA. DNA is present within the nucleus of a cell and DNA is organized into chromatin. So to see DNA, that is in turn the chromatin, we are going to use a stain. Here the stain we are using is acetocarmine or we can also use acetoarsine. So these stain are basic stain which has an affinity to bind with acidic components. Here the acidic component is DNA. You all know that DNA is acidic in nature because of the phosphate group present in it. So the acidic DNA can bind with the basic stain acetocarmine. So the acetocarmine or acetoarsine, these are mostly used to stain the chromosomes because they are chromatin specific dyes and they readily color the acidic component, that's nothing but DNA, which is present inside the nucleus. So in this experiment, we are going to mainly stain the DNA. Okay, now let us start with the experiment. So take a piece of onion scale leaf. And here the leaf will be having a upper epidermis and lower epidermis. So upper epidermis you could differentiate with slight pink color. So for the experiment we are not taking the upper epidermis which is slightly pink. We have to take the colorless one, the transparent one which is the lower epidermis for the experiment. So using a forceps, we will just peel out the lower epidermis of the onion scale leaf. So just if you peel off with the forceps, you will get a transparent epidermal peel and make sure that the peel should not get dry. So you have to place it on a clean glass slide and using a brush, take a just drop of water and then spread the peel without any foldings. So the peel should not have any foldings. It should be clear. Then we are going to cut the peel in a size that the size should be within the area of the cover slip. The excess one can just take it out. Make sure there is no air bubble present on the peel. Just clear it using a brush and the peel should be within the area of the cover slip. So now the peel is ready and now to this peel I am going to add 2 drops of acetocaramine stain on the material. And now just spread the stain uniformly on the peel so that there will be no air bubbles as well as the material gets stained properly. So this can just continue to do for about 30 seconds or 1 minute so that it gets stained properly. This to be done using a brush. Now, after staining the material, the excess stain we are going to just wash it out. Rather blotting it, it's better to remove the stain like this using the water. Just with the help of brush, drain the excess stain with water. Now here the onion peel is now stained and it is ready to focus. So now to this we will place the cover slip. So you all know that to place the cover slip you will be using a dissection needle. Place the cover slip oblique to it and gently cover the material. Yes, now the slide is ready to focus. So now the slide what we have prepared, we are going to focus it under 10x and we are going to see the shape of the cell, and the main aim of the experiment is that we have to observe the nucleus. So that for that we have stained using a acetocaramine. So now I am focusing the slide under 10x to see the nucleus. Yes, now I am able to see the cells with a spherical stained nucleus. So the cells are appearing rectangular in shape, and each cell will have one centrally located nucleus, and the nucleus is appearing pink in color. That is due to the stain what we have used. Now coming to the end of the experiment, as I told earlier, the preparation of the slide carries 3 marks and the result carries 2 marks. So while you write the result, you have to report 2 important observations, that is the shape of the cell observed. So as we have taken the epidermal peel, and you all know that the shape of the epidermal cell is rectangular. Or you can also write it as polygonal in shape. Rectangular or polygonal in shape. And then the second observation is that the number of nuclei present in each cell. So each cell contains one nucleus that is centrally located and that is stained by acetocarmine, and while we see through the microscope it appears pink in color. So we report that the number of nuclei present in each cell is 1. So these are the 2 important observations that you have to report while you see through the microscope.